Recording. Share the screen. Okay. If there's no questions, let's go ahead and begin. So we're halfway through. It's been a wild ride and we're moving very quickly, meeting 4 times a week. It always does that in the summer. We're on chapter 13. Let me remind you that we changed the lab schedule. I moved lab 9 up a week, and that will give you more time to work on your unknown project. Let me remind you, you have a quiz on Friday this week. You'll have to have the respondents lockdown browser. I haven't made the quiz yet, so it's not showing that, but you'll have to have that. The quiz will be over chapter 8 and lab 4. Any questions? If not, this is the last slide we were talking about last time. So we're talking about the viral multiplication cycle, and in bacteriophages. Sorry? Can somebody repeat that? I didn't hear that. Let me turn the volume up. Was somebody asking a question? All right. If there's no questions, we're talking about the viral multiplication cycle. And in bacteriophages, they have 2 life cycles. There's the lytic life cycle and the lysogenic life cycle. We'll discuss the lytic life cycle now. The lytic life cycle has 5 stages. The virus has to have attachment and then go through penetration, biosynthesis, maturation of viruses, and then release. In attachment, the phage attaches to the host cell that the virus replicates in, and that's generally only one species of bacteria for most lytic viruses. The phage, when I say phage, I mean bacteriophage, sorry. The bacteriophage then penetrates the host cell, and it actually secretes lysozyme. That's an enzyme we will talk about later. I don't remember if we've talked about that yet. I don't think we have. That's an enzyme that opens up the cell wall. It damages the cell wall of gram-negative and gram-positive cells. It digests the peptidoglycan. Anyways, the virus secretes lysozyme, opening the cell wall, and then the tail sheath literally contracts, sort of like a syringe contracts to inject the serum into your blood. Well, the tail sheath contracts and it injects the nucleic acid in the head of the virus into the cell, the bacteria cell. And that then infects the DNA of this virus, the T4, into the cell. The virus then replicates itself, so it makes new phage DNA, and it transcribes the viral DNA, making viral messenger RNA, which will be translated into viral proteins. And then the virus, the new virions, reassemble in maturation, where the viral proteins get together to make the virus, and the viral DNA is put inside the viral capsid coat, and then the new virions are assembled. And then lastly, the last thing the virus does in the lytic life cycle is it destroys the cell wall, causing the cell to lyse, and that will release the new virions. How the viruses do that is they secrete the enzyme lysozyme, and that destroys the cell wall of the virus, and fluid moves into the virus, and then the host cell rupts, the cell membrane expands, and then it just ruptures, and that releases the new viruses. Let's watch a little video. Let's see if I can get this going on the lytic life cycle.
In a way, a virus is a parasite. It attacks.
Are you hearing that?
Yes.
Okay. Thank you. Let me blow that up.
The host cell and takes over its chemical machinery for making proteins and nucleic acids. One of the better understood virus life cycles is phage, a virus which attacks bacteria cells. The phage begins its assault by attaching itself to the cell wall of the bacteria. This attachment is very specific, and each type of phage can only attach to a certain type of bacteria. Next, the phage breaks down the cell wall, and the nucleic acid from this capsule is injected through the hollow tail into the cell. The viral nucleic acid uses the cell's enzymes and organelles to replicate new viral parts. The parts are assembled into new virus particles called virions. Finally, the new phages release an enzyme which weakens the cell wall, and the host cell bursts, releasing the virions. Up to 300 new virus particles can be reproduced in a single host cell.
Okay, any questions about that? That took you through the stages of the lytic life cycle. I'm going to keep that movie up because the next one is on the next life cycle. But let's go through the lytic life cycle in great detail. The virion will attach, a virus will attach to the cell wall of a bacteria, and like I said, it digests the cell wall right here, and then the virus literally contracts, meaning the sheath contracts, and that puts pressure on the head, and that injects the DNA of the virus into the cell. The DNA of the virus then replicates itself, making more DNA of the viral DNA, and one of the early things the viral DNA does, even before it replicates the new viral DNA, is it secretes an enzyme that destroys the host chromosome. And that way the genome of the virus is running the cell. So by destroying the host chromosome, you allow your genome to run everything in the cell. And then the viral DNA is transcribed and translated, and that makes the proteins of the virus, and it's trying to show you the head proteins, the sheath proteins, and the tail fiber proteins. And then the new virions are assembled, meaning that the different proteins come together in the shape of the new virion. And it doesn't show that here, but before the virus is finalized, the DNA of the virus is packaged into the heads of the virus. and then that head is attached to the sheath, and the sheath is attached to the tail fibers, making the new virion. And then there's one copy that isn't of the virus, that isn't put into the new virions, and it is transcribed, and what it does is it secretes the gene which makes an enzyme that weakens the cell wall of the virus. And then the cell wall bursts, and that releases the new virions. Any question about the lytic life cycle? There's two terms you should know in the lytic life cycle. That is the burst time. That is the time from when virion first infects a host bacterial cell till the time the cell bursts, releasing the new virions. Okay? And the burst time will depend on the temperature you're doing this at, but at standard temperature, the average time for, or the average burst time for the viruses are 20 to 40 minutes, depending on the viral species. And the other term to know is the burst size. That's the newly replicated virions that are released when the host bacteria cell bursts. Once again, that will depend upon the virus. It also depends on how fortunate the virus is and replicating itself inside the cell. And I imagine that has to do with how many resources the cell has. But generally, between 50 new virions can be made, and it can be over 200, but the average is 50 to 200, and it can go up to 300. Any questions on the burst time or the burst size or the lytic life cycle? Okay, some bacteriophages only have the lytic life cycle, and that is how they replicate. And I can tell you there are some human viruses that only replicate with the lytic life cycle. So they will infect your cells, replicate inside your cells, and then lyse your cells to release the new virions. Any question about any of that? All right, the lytic life cycle isn't the only life cycle that bacteriophages can have. They can also have the lysogenic life cycle, where the virus does not kill the host cell, at least doesn't kill the host cell immediately. So what happens in the lysogenic life cycle is there will be an attachment of a bacteriophage, and then penetration of the cell wall by the virus, injecting the viral genome into the cell. We're only going to talk about it with the DNA viruses, and we're going to talk about it happening with the bacteriophage T4. And why we're going to talk about it with the bacteriophage T4 is that this virus is the most studied of all viruses. It's more studied than any human virus. And I think the reason why it's simply the most studied virus is that bacteriophage T4 infects E. coli cells, and that's a common cell in a microbiology lab. So bacteriophage T4 is the most studied virus. And when a lysogenic virus infects a host cell, and that the viral DNA is infected into the host cell, the lysogenic virus has to decide immediately what life cycle it wants to enter. It can enter the lytic life cycle, or it can enter the lysogenic life cycle. If it enters the lytic life cycle, the life cycle we just talked about, where the virus will start replicating. It'll destroy the host chromosome. It'll be transcribed and translated. New virions will be made, and they'll assemble, and the viral DNA will be replicated. And it'll then lyse the cell, releasing the new virions. And that is the lytic life cycle of a lysogenic virus. But as I stated, the lysogenic virus, when it first infects a host cell, it decides whether to enter the lytic life cycle or the lysogenic life cycle. If it enters the lysogenic life cycle, there's a gene of the lysogenic life cycle, which is a repressor protein, and it represses the genes of the lytic life cycle. Any question about that? And then the lysogenic viral DNA inserts itself into the DNA of the host chromosome. And that begins the lysogenic life cycle. The phage remains inactive. You could call it latent, because the lytic genes of the lytic life cycle are repressed. The only time the virus replicates when it's inside the host cell's DNA is when the host cell itself replicates its DNA and then divides. And the bacteria and the host remain together in symbiosis in the lysogenic life cycle. And the virus is termed a prophage, is that right? Anyways, the host cell remains alive in the lysogenic life cycle, and the viral DNA is part of the chromosome to the host cell in the lysogenic life cycle. Let's look at that in a little more detail. So the lysogenic virus infects this host cell, and immediately it decides, does it enter the lytic life cycle, or does it enter the lysogenic life cycle? I've already talked about the lytic life cycle. It's just the same thing we referred to. But if it enters the lysogenic life cycle, the viral DNA integrates into the host chromosome, and it's called the prophage. The only time the viral DNA replicates is when the host goes through cell division, and it'll replicate its DNA, and that will replicate the lysogenic viral DNA. And this can go through many cell cycles of that. Okay? Any questions about the lysogenic life cycle?
Does there ever come a time when a cell that's in the lysogenic cycle ever turns its enzymes back on and goes back into the lytic cycle?
The lytic life cycle, yes it does. What happens is sometime later in the bacteria's reproduction, on one of the cells, it doesn't have to happen on all of them, usually it's just one of the cells, the viral DNA excises itself from the host chromosome. And now we're like this. And the repressor protein stops functioning or gets blocked, so it's no longer active. And then the lytic genes of the virus become expressed, and then we enter the lytic life cycle. Okay? And what switches the cell from leaving the lysogenic life cycle to entering the lytic life cycle is not well understood. And they've been studying this now for 70 years, and they still don't understand what switches the lysogenic life cycle to the lytic life cycle. They do know that one thing that can switch the virus from leaving the lysogenic life cycle to the lytic life cycle is if something damages the DNA of this cell. That tends to switch the cell from the lysogenic life cycle to the lytic life cycle. But sometimes that switch happens and we don't have a good understanding for why that switch happens. Okay? Any questions about that? I think I've already talked about this. I've talked about the repressor protein X keeping the cell in the lysogenic life cycle, and what happens when the cell switches to the lytic life cycle, the repressor protein stops acting. It might get blocked. All right, let's watch the movie on the lysogenic life cycle. And I've already got that right here.
This process of viral multiplication is called the lytic cycle. Interestingly, the lytic cycle does not always happen immediately. Sometimes, rather than producing virions, phage nucleic acid incorporates in the host cell DNA. Now, each time the cell divides, the phage DNA is also reproduced and is spread to more and more cells. Then suddenly, for reasons that are still unclear, the phage DNA activates, and the destructive lytic cycle begins. Once again, the host cell is destroyed, and a new generation of phage are released to invade new hosts.
All right, any questions about this movie? Come on. Everything is correct in this movie except for one thing. And this is showing you bacteriophage T4. Bacteriophage T4 only has the lytic life cycle. It doesn't go through the lysogenic life cycle. Okay? Everything else about this movie is right on. No, don't want to do that. How do I get rid of that movie? There we go. There are some consequences for lysogeny. A lysogenic cell, while it has the prophage in it, meaning the viral DNA is integrated into the host cell's DNA, this cell is immune to infection by the same species of virus. So another lysogenic virus will not infect this cell. So this cell is immune to infection by the lysogenic virus. And that's because it already has the lysogenic virus in it and is following the lysogenic life cycle. Any question about that? Another consequence for lysogeny is that phage conversion may occur, and that is the host cell can express a gene of the virus, changing the phenotype of this cell. Any questions about that? So the viral DNA can bring the host cell a new gene which can change the phenotype of the bacteria cell. And that's called phage conversion. An example of that is Corynebacteria diphtherium is a bacteria that produces diphtheria toxin. Well, the diphtheria toxin is only produced in this bacteria when the bacteria is infected with a lysogenic virus, meaning the lysogenic virus has the gene that produces the Corynebacterium diphtheria toxin. So if you're infected by Corynebacterium diphtheria that doesn't have a lysogenic virus, you won't be exposed to the diphtheria toxin. Any questions about that? Another consequence is specialized transduction, and I'm not going to talk about that. You can read about it in the textbook if you want. I'm not going to hold you to specialized transduction. We've talked about generalized transduction, and the specialized transduction is similar. That's where a virus can bring a cell new DNA, but I'm not going to talk about specialized transduction. Any questions about the consequences of lysogeny or the lytic life cycle? Okay. If not, let's move on then. We've talked enough about bacteriophages. They only have 2 life cycles. Now we want to talk about animal viruses. What happens in the multiplication of animal viruses? So when we're talking about the multiplication of animal viruses, they follow the basic pattern for bacteriophage multiplication, but they have a few differences. For example, when viruses attach to their host cell, an animal virus doesn't attach to the cell wall because they have to attach to the cell membrane. Animals don't have a cell wall. So the attachment's a little different, but essentially it's the same. The virus has specific attachment sites that bind to receptors on the host cell, so that's the same. The penetration is a little different. It's generally easier to get inside an animal cell than it is a bacteria cell. There's no cell wall to get through, for one thing. And penetration of an animal virus happens in 2 ways. It either happens by endocytosis, I'll talk about that a little more, or by fusion. If the animal cell has a lipid envelope, the lipid envelope can literally fuse to the cell membrane, and then the 2 become 1, and the virus is just dumped into the cytoplasm of the cell. And I'll show you a little picture of that. Uncoating can be by viral or host enzymes. We didn't really talk about that for bacteriophage, so no change because we're not talking about it. Biosynthesis is still the same. You have the production of the nucleic acids and the proteins, and maturation is similar. There is one difference in maturation, and that is that in animal viruses, I'll talk about that, they have a choice as to where to replicate. In a bacteriophage, they only have one place to replicate, and that's the cytoplasm. And animal viruses can replicate in the cytoplasm, but they can replicate in the nucleus. Okay? Any questions about that? And then release can be the same. Like if we have a lytic animal virus, it can lyse the cell and the cell ruptures, so that can be the same. But we also have a budding virus, and that generally does not kill the cell. So the release of the virus can be different depending on what life cycle the virus is following. All right. For the differences for animal viruses and bacteria viruses, the main difference is that you should realize that animal viruses can replicate in the nucleus or the cytoplasm. Bacteriophages only replicate in the cytoplasm. Any questions about that? There is a difference of how the virus also attaches to the cell and then enters the cell. With animal viruses, they attach by endocytosis, and the virus, when it attaches to the cell, it causes the cell to invaginate at that region, and the invagination grows, and then this breaks into the cytoplasm as a vesicle, and the cell membrane reattaches right here, and then it reattaches right here to make the membrane of the vesicle, and then this vesicle moves into the cell. And so that's how the virus moves into the cell by endocytosis. Any question about any of that? Let me see if I've got fusion. Not really. With fusion, we have a viral envelope, and the viral envelope combines with the viral cell membrane, and so the membrane, instead of coming across like that, it now comes around like this, and this picture's sort of showing it, and that will dump the virus into the cell. That's really showing you endocytosis, not fusion. But when the cell membrane then comes around like that, this opens up, and that allows the virus just to move into the cytoplasm of the cell. Any questions about that? So the encoding of the virus can happen by viral or host enzymes. The biosynthesis happens similar to what happens in a bacteriophage. The nucleic acid of the virus will be replicated. It will be transcribed and translated into proteins. And then the new virions will assemble, and that we call maturation. There is one difference, and that is animal viruses can lyse the cell and be released by the lytic life cycle if the virus goes through the lytic life cycle. But not all animal viruses follow the lytic life cycle. There are also budding viruses, meaning a budding life cycle. Animal viruses that don't lyse the cell, they enter the budding life cycle. What happens with this one is the viruses replicate slowly, meaning they don't replicate hundreds of viruses in the cell at the same time. Usually they replicate slowly, and then when the new virion is made, it comes up to the cell membrane or another membrane. It doesn't have to be the cell membrane. It could be the nuclear membrane or the membrane of the endoplasmic reticulum. We're showing you for the cell membrane. And it'll come up to the cell membrane where the virus has made proteins and glycoproteins, which are put in the cell membrane. So, and if you notice on this part of the cell membrane, it doesn't have viral proteins and glycoproteins. Well, the virus doesn't come up to this region. Sorry, I lost my mouse. The virus doesn't come up to this region of the cell membrane. It comes up to this region of the cell membrane where it has proteins and glycoproteins that the virus made synthesized, and then stuck those proteins and glycoproteins into the cell membrane. And then it pushes on the cell membrane, forming a bud of the cell membrane. And then it rips off a bud of the cell membrane, or the nuclear membrane, or the membrane of the endoplasmic reticulum. Like I said, we're showing you for the cell membrane. And that is how a budding virus obtains its envelope. This envelope is largely the phospholipid bilayer of a membrane of the cell, such as the cell membrane. Everyone see that? And this cell, the membrane, reforms, and generally this cell does not die from the budding virus. So this cell can over time bud off many viruses. If this cell dies, it's because the host immune system, usually it's because the host immune system recognizes that it is infected with a virus, and then the host immune system kills this cell. The budding virus, meaning a virus that follows the budding life cycle, generally does not kill the host cell it's infected. And here we're seeing an actual bud in an electron microscopic image. And there are several buds that you can see on this cell. Any questions about the budding life cycle? And that is how animal viruses obtain their envelope, if they have an envelope around the virus. Any question about that? All right, so when viruses have a different genome, they have to use a different strategy for replicating their genome. So we'll talk a little bit about that. The DNA viruses are very easy to explain how they replicate their viral genome, and that is double-stranded DNA just separates a single strand DNA, and then one strand is used as a template to make the complementary strand of DNA. And then we replicate our double-stranded DNA virus. Any question about that? That just follows the same way the host cell replicates its double-stranded DNA. Single-stranded DNA virus is a little different. The single-stranded DNA will use that genomic single-stranded DNA to make the complementary strand of DNA, meaning if this is, what would it be, CCT, then it would make GGA as the complementary strand. You got that? And then the complementary strand of DNA is used to make the genomic viral DNA. Okay? For double-stranded RNA viruses, it's pretty simple. You just separate the double-stranded RNA and make it single-strand RNA. And then you use one strand of the RNA as a template for making the other strand of RNA. So it's very similar to double-stranded DNA, it's just we're using RNA. But that's how a double-stranded RNA virus replicates its double-stranded RNA genome. And each strand is replicated to make the other strand. And then the RNA, the double-stranded RNA genome is replicated. Any question about that? I wonder if I want to talk about the single-stranded RNA. Let me look, because I do talk about it, but I don't talk about it until later. The single-stranded RNA, we'll talk about it more later, but what you do is you read the single-stranded RNA genome to the virus, and you make the complementary strand of RNA. And then you use that complementary strand to make more viral single-strand RNA genome. Is that clear? I'll talk more about that later, but I don't want to talk about that in great detail here. Because this one is, well, it's just complicated, okay? But it follows the same pattern as single-stranded DNA, and that is you make a complementary strand to the single-strand RNA, and then you use the complementary strand RNA to make your genomic single-strand RNA. So each virus type needs to have a slightly different mechanism for multiplication and biosynthesis. Let's talk about the DNA containing animal viruses. For DNA containing genomic viruses, for animal viruses, the DNA viruses generally are replicated in the nucleus of an animal cell. The reason why that is because all of the enzymes that the virus needs to replicate itself can be found in the nucleus of an animal cell. Like DNA polymerase is found there if the virus needs it to replicate its DNA genome. And then all the other things like the nucleotides, ATP, what else is required? Helicase, things like that. And then the viral genome is transcribed, and the messenger RNA is moved into the cytoplasm. And in the cytoplasm is where the capsids and the viral proteins are synthesized. And why that is, and meaning the virus replicates its proteins in the cytoplasm of the animal cell, is that's where all the ingredients for an animal cell, where the ingredients are for the virus to replicate all of its proteins. That's where the ribosomes are, that's where the tRNA is, that's where the amino acids are. Okay, so all the enzymes and machinery that the virus needs for replicating the proteins of an animal cell are found in the cytoplasm. Any question about any of that? Once a animal viral DNA enters the cell, the viral DNA will then enter the host nucleus. One of two things can happen. The DNA can enter the viral DNA can enter the DNA of the host cell. If that happens, the viral DNA permanently enters that host cell's chromosome, and it will never be excised in an animal cell, and that will remain for as long as that cell is alive. So if the DNA enters the cell and then enters into the host cell's DNA, that viral DNA becomes a permanent copy of that host cell's DNA. It's never excised in an animal cells. And the only, well, that's not true. And the viral DNA will be replicated when that host cell replicates its DNA and then goes through cell division. Any questions about that? What we call the viral DNA when it's in the host cell's DNA, we don't call it a prophage because this is not a bacteriophage. It's not a bacteria virus. It's an animal virus. So it's called a provirus. So if you hear the term provirus, that's where the viral DNA is integrated into the host cell's DNA. I'm trying
Is that
to
what
think
makes
of
some viruses harder to get rid of?
well, that will become permanently into that host cell's DNA. So yes, it's hard to get rid of. The only way to get rid of it is the host immune system would kill this cell. Okay? It is possible that the virus, when it's replicating, could kill the cell, but generally it'll be killed by the host immune system. It'll somehow recognize that this cell is infected and then kill it. Otherwise, this cell will continue, perhaps permanently, in the animal. If the host cell immune system doesn't have it, that cell can survive for as long as the cell lives. And that could be for the lifetime of the animal. A viral DNA that can do this is the viruses that, papillomaviruses, viruses that cause warts. Okay? And papillomavirus can also cause tumors. Anyways, that's a provirus, where the viral DNA can integrate into the host cell's chromosome. The other thing that can happen when the viral DNA enters the cell, the viral DNA can be immediately expressed, producing the viral proteins, and then the viral DNA can be replicated. So the viral DNA can be copied, the viral DNA can be transcribed and then translated, new capsids can be made, new virions can be made, and then this cell, if it's a lytic virus, it can lyse the cell and release the virions. If it's a budding virus, it'll bud out of the cell, and that won't lyse the cell. So let's look at this slide. In general, this is a lytic life cycle. The virus infects the cell, and first it attaches to the cell membrane, and then the virus is brought into the cell by either endocytosis, or if it has an envelope, by fusion of the envelope's membrane with the cell membrane. Anyways, the virus gets into the cell, the viral DNA is read, transcribed, and translated. The viral DNA has to move into the nucleus of the cell because DNA viruses replicate in the nucleus of the cell. And the viral DNA can be transcribed. The messenger RNA, the viral messenger RNA, comes into the cytoplasm, and they will be translated into the viral proteins, such as the capsid proteins. In the meantime, the viral DNA in the nucleus can be replicated, and then the viral DNA can assemble into the new virions. They'll get together with the capsid proteins, making the functional new virions. And then in the lytic life cycle, the cell lyses the cell, and that will release the new virions. Any questions about a lytic DNA virus of an animal cell? Okay, if there's no questions, let's move on to the next slide. We'll just briefly talk about the RNA viruses, and then we'll stop for the class. I think it ends at 2:15, is that right?
Yes.
Yeah, all right. Thank you. I'm just going up here to show you. And then we'll come back for our next session. Um, so, did I show you a table of DNA viruses? Well, that's a mistake. Why didn't I show you a table of DNA viruses? Oh, yes, I did. Here are the different families of DNA viruses. I know you can't see this. Let me blow it up as best I can. You don't need to know the families. I'm just showing you that there are a number of different viral families which are DNA virus that infect animal cells and they cause different diseases. Here's the herpes virus or herpes viridae. We've talked about those. I briefly mentioned the papilloma viruses. Adenoviruses, that's a virus that can cause some of the human colds. You probably don't know that one. Anyways, those are just some different DNA animal viruses. Did I talk about that slide? I think I went past it.
Yeah, you went past it.
Yeah. Sorry about that. Here is the slide of the animal viruses that are RNA viruses. And as you notice, let me blow that up a little bit. If I blow it up more, you're not going to see all the animal viruses. But there's more of them, okay? So there's more families of RNA viruses. I believe there's 15 of them that infect animal cells. And some of them are pretty important. We're not really going to talk about the different viruses, but Picornaviridae, that's the #1 cause of the colds, the rhinoviruses, excuse me, cause colds. Picornaviridae is also the family that has the poliovirus. You probably don't know Astroviridae and Togoviridae, but if you heard of like Eastern equine encephalitis and what's the other one? Western equine encephalitis, they're in those families. Rubiola is a Togoviridae. You've probably heard of that virus, the German measles. I'm trying to get my mouse to work. Coronaviridae, I'm sure you've heard of that. That's the coronaviruses, and COVID-19 belongs to this family. So we'll talk about this family later in the lesson. And then Retroviridae, that's the retroviruses. They're different from all the other RNA viruses, so we'll talk a little bit about that family of viruses. The paromyxoviruses, cause colds and respiratory symptoms like pneumonia and the flu. So that's an important family of virus. We won't talk about the orthomyxoviridae that cause the flu. And that's the last one that I just wanted to briefly talk about. Anyways, the point is there's lots of RNA families that infect human cells, and let's talk just briefly about the animal viruses. Here you're looking at a couple of the RNA viruses, and you can tell immediately that they have different shapes. Obviously, they're different names. The multiplication of RNA viruses happen generally similar to the DNA viruses, but there are some differences because they have an RNA genome instead of a DNA genome. The main difference you need to know about is that the RNA-containing viruses replicate in the cytoplasm, not in the nucleus. The DNA viruses, animal viruses replicate in the nucleus, but RNA animal viruses replicate in the cytoplasm. That is the main difference. And that rule of thumb holds for everything except, there is one exception, the retroviruses do not follow this rule of thumb. And we'll talk more about the retroviruses later. They are an RNA virus, but they actually replicate in the nucleus, not in the cytoplasm. And that's the only one that's the exception to that rule of thumb. There are some other differences that you need to know about when we're talking about RNA viruses and how they replicate. Obviously, they're RNA for a genome, so there are some differences. For the double-stranded RNA viruses, you just use one strand to make a copy of the other strand. Okay? So the double-stranded RNA viruses replicate essentially similar to double-stranded DNA. It's just that you're using RNA instead, and you're making RNA instead of DNA. There are some differences, though, when we're talking about single-stranded RNA viruses. And when we're talking about an RNA virus that's single-strand, there are two types. There's the positive strand, single-strand RNA virus, and there's the negative strand, single-strand RNA virus. A positive strand, single-strand RNA genome can be read by the ribosomes. So this genome for the RNA animal virus can be read by ribosomes to translate into protein. Any question about that? So this genome of the virus serves as both the genome of the virus and the viral messenger RNA. So if we have AUG, the ribosome can read that and translate it into proteins. The negative strand RNA animal virus cannot be read by the ribosomes. It's the antisense to the positive strand. So instead of having AUG, it will have UAC, the complement to the positive strand. And the ribosomes can't read that. Any question about that? So this genome is only the genome. And to make viral RNA, it will copy the negative strand and make the corollary, which is the positive strand. And then that positive strand RNA can be used as viral messenger RNA. Any questions about that? Let me show the slide here. So if we have a DNA virus, you just transcribe the DNA to get the viral messenger RNA and translate that into protein. That's pretty standard. If you have, oh, that's the positive strand RNA, the messenger RNA. If you have an RNA strand virus, positive strand virus, to replicate the positive strand of the viral genome, you make the antisense negative strand RNA, and then you use this negative strand RNA to make the positive strand RNA genome. And that's how a positive strand replicates its genome. It makes the negative strand RNA, which is the corollary or antisense, and then it uses that to make more of the positive strand viral genome. The negative strand is not read to make protein. Okay? If we have a negative strand RNA viral genome, it will make the positive strand RNA complementary to the negative strand, and it can use that positive strand RNA as messenger RNA for the virus and translate it into the viral protein. Any questions about that? That positive strand RNA for the genome, which is negative strand, can then be used to replicate more negative strand RNA genome of the virus. So if you have this strand, you make that strand, then you use this strand to make more of this strand. Any questions about that? And that is how negative strand RNA virus replicates itself. Any questions about how viruses replicate and how they translate viral RNA into viral protein? This is just going through it one more time. If you have negative strand RNA genome, you make the positive strand, and then you use the positive strand to make more negative RNA strand of the virus, and this will be the viral genome. Use this strand to translate it to make the capsid viral proteins, and then you get the capsid proteins with the negative strand to make the new virions. It's a little easier for the positive strand RNA. You make more negative strand RNA, use that strand to replicate the viral genome, positive of the genome, and then you use this strand to translate the viral proteins, and then you get the capsid proteins together with the viral genome to make the new virions. Any questions about any of that? Do we have time to talk about this? Yes. All right. So this is a positive sense single-strand RNA virus. Let's talk about how it infects a cell and then replicates. So a new virus that is a positive strand RNA infects the cell first by attaching to the cell, and then it can come into the cell by endocytosis, or if it's an envelope virus, which I don't think that's showing you an envelope there, the envelope can fuse to the cell membrane, and then once that happens, the virus can be put into the cytoplasm of the cell. Now this is an RNA virus, so it stays in the cytoplasm and replicates in the cytoplasm. It's a positive strand, so that positive strand RNA can be read by the ribosomes to make viral protein. And my mouse died. Let me try and get the mouse back. And so that positive strand can be used as messenger RNA by the ribosomes to make viral protein. And it's already in the cytoplasm, so there's no trouble there. And then the viral positive strand RNA makes negative strand, single strand RNA, and then that strand is used to make more of the positive strand RNA, which is the genome of the virus. And that happens in the cytoplasm because RNA viruses replicate in the cytoplasm. And then the genome of the RNA virus gets together with the proteins of the virus, making new capsid proteins. And these viruses are released in the lytic life cycle. Come on, mouse. This cell will lyse, and that will release the new virions. If this cell is a budding virus, that virus will be released by budding. Meaning, in this case, it will come up against the cell membrane, rip off a part of the cell membrane, and that'll become the envelope of the budding virus. Any question about any of that? For the negative strand RNA virus, I'm just going to talk about this quickly because it's similar. The virus attaches in the same way, enters the cell in the same way, and then begins replicating in the same way in the cytoplasm. The viral genome is a negative strand, so it cannot be immediately translated or transcribed and translated. It first has to make positive strand. I think it changed color on me here. It looks like that's the negative now. Oh, I see. One's making that one. So let's use this one because that's following the same color. The viral genome is negative strand, and it makes the positive strand RNA, which can act as viral messenger RNA. And so this strand can be read by the ribosome to make the viral proteins. And then this positive strand is used as a template to make more of the viral genome. And so we have the positive strand right here, and then that's used as a template to make more of the negative strand, which is the genome of this RNA strand because it's a negative strand. And then the genomic RNA gets together with the viral proteins to make the new virions. And then that virion will be released from the cell either by lysis of the cell or by budding, depending on what life cycle the virus has. Any question about that? All right. I think I'll talk about retroviruses next time. So we're going to break just a few minutes early, and I'll see you at 2:30 for the second part of the lab. Actually, the lecture today. Any questions? If there's no questions, I'll see you at 2:30.